Biochemistry, laboratory techniques. Polymerase chain reaction. Polymerase chain reaction is a molecular biology lab procedure used to amplify a desired fragment of DNA. It is useful as a diagnostic tool, for example for diagnosing neonatal HIV and herpes encephalitis. 1. Denaturation. DNA template, DNA primers, a heat-stable DNA polymerase, and deoxynucleotide triphosphates are heated to separate the DNA strands. 2. Annealing. The sample is cooled. DNA primers anneal to the specific sequence to be amplified on the DNA template. 3. Elongation. The temperature is increased. DNA polymerase adds deoxynucleotide triphosphates to the strand to replicate the sequence after each primer. Heating and cooling cycles continue until the amount of DNA is sufficient. CRISPR-Cas9. CRISPR-Cas9 is a genome editing tool derived from bacteria. It consists of a guide RNA, which is complementary to a target DNA sequence, and an endonuclease Cas9, which makes a single or double-strand break at the target site. Applications of CRISPR-Cas9 include removing virulence factors from pathogens, replacing disease-causing alleles of genes with healthy variants, for example, sickle cell disease, and specifically targeting tumor cells. Blotting procedures. Southern blot. 1. DNA sample is enzymatically cleaved into smaller pieces which are separated by gel electrophoresis and then transferred to a membrane. 2. Membrane is exposed to labeled DNA probe that anneals to its complementary strand. 3. Resulting double-stranded, labeled piece of DNA is visualized when membrane is exposed to film or digital imager. Southern blot is useful to identify the size of specific sequences. For example, determination of heterozygosity and number of CGG repeats in Fragile X messenger ribonucleoprotein 1 to diagnose Fragile X syndrome. Northern blot. Northern blot is similar to southern blot except that an RNA sample is electrophoresed. Northern blot is useful for studying messenger RNA levels and size, which are reflective of gene expression. Northern blot also detects splicing errors. Western blot. In Western blot, sample protein is separated via gel electrophoresis and transferred to a membrane. Labeled antibody is used to bind relevant protein. This helps identify specific protein and determines quantity. Southern DNA, northern RNA, western protein. Flow cytometry. Flow cytometry is a laboratory technique to assess the size, granularity, and protein expression of the individual cells in a sample. Protein expression is the immunophenotype. Cells are tagged with antibodies specific to the surface or intracellular proteins. Antibodies are then tagged with a unique fluorescent dye. Eventually, sample is analyzed one cell at a time by focusing a laser on the cell and measuring the light scatter and intensity of fluorescence. The data are plotted either as histogram, one measure, or scatter plot, any two measures, as shown in the illustration. Cells in the left lower quadrant are negative for both CD8 and CD3. Cells in the right lower quadrant are positive for CD8 and negative for CD3. In this example, right lower quadrant is empty because all CD8 expressing cells also express CD3. Cells in the left upper quadrant are positive for CD3 and negative for CD8. Cells in the right upper quadrant are positive for both CD8 and CD3. Flow cytometry is commonly used in the workup of hematologic abnormalities, for example, leukemia, paroxysmal nocturnal hemoglobinuria, fetal red blood cells in pregnant person's blood, and immunodeficiencies, for example, CD4 cell count in HIV. Microarrays. Microarrays are used to compare the relative transcription of genes in two RNA samples. Microarrays can detect single nucleotide polymorphisms and copy number variants for genotyping, clinical genetic testing, forensic analysis, and cancer mutation and genetic linkage analysis when DNA is used. Enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay is an immunologic test used to detect the presence of either a specific antigen in direct ELISA or antibody in indirect ELISA in a patient's blood sample. The detection involves the use of an antibody linked to an enzyme. The added substrate reacts with the enzyme, producing a detectable signal. ELISA can have high sensitivity and specificity, but is less specific than Western blot. ELISA is often used to screen for HIV infection. Karyotyping. In karyotyping, colchicine is added to the cultured cells to disrupt the assembly of mitotic spindles and arrest the cells at mitosis. The chromosomes are stained, ordered, and numbered according to the morphology, size, arm-length ratio, and banding pattern. Arrows in A point to extensive abnormalities in a cancer cell. Karyotyping can be performed on a sample of blood, bone marrow, amniotic fluid, or placental tissue. Karyotyping is used to diagnose chromosomal imbalances, for example, autosomal trisomies, sex chromosome disorders. Fluorescence in situ hybridization. In fluorescence in situ hybridization, fluorescent DNA or RNA probe binds to the specific gene or other site of interest on chromosomes. Fluorescence in situ hybridization is used for specific localization of genes and direct visualization of chromosomal anomalies. Microdeletion. Microdeletion is no fluorescence on a chromosome compared to fluorescence at the same locus on the second copy of that chromosome. Translocation. Translocation is fluorescence signal that corresponds to tetrasomy, gain of chromosome, and unbalanced translocation. Duplication. Duplication is a second copy of a chromosome, resulting in a trisomy or tetrasomy, prosomy, and posomy. Molecular cloning. Molecular cloning is production of a recombinant DNA molecule in a bacterial or eukaryotic cell line host. Molecular cloning is useful for production of human proteins in bacteria. For example, human growth hormone, insulin. Gene expression modifications. Transgenic strategies in mice involved. Random insertion of gene into mouse genome. Targeted insertion or deletion of gene through homologous recombination with mouse gene. Knockout is removing a gene, taking it out. Knock-in is inserting a gene. Random insertion leads to constitutive expression. Targeted insertion leads to conditional expression. RNA interference. RNA interference is a process whereby small non-coding RNA molecules target messenger RNAs to inhibit gene expression. Microarray. Microarray is naturally produced by cell as hairpin structures. Loose nucleotide pairing allows broad targeting of related messenger RNAs. When microRNA binds to messenger RNA, it blocks translation of messenger RNA and sometimes facilitates its degradation. Abnormal expression of microRNAs contributes to certain malignancies, for example, by silencing in messenger RNA from a tumor suppressor gene. Small interfering RNA. Small interfering RNA, usually derived from exogenous double-stranded RNA source, for example, virus. Once inside a cell, small interfering RNA requires complete nucleotide pairing, leading to highly specific messenger RNA targeting, results in messenger RNA cleavage prior to translation. Small interfering RNA can be produced by transcription or chemically synthesized for gene knockdown experiments